Welcome, everybody, once again. Thank you very much for joining us in this virtual event. This is now the second part of the Oncomine World Autumn event, and now we will move on to another section. We'll have two more speakers now. The first one is Dr. Javier Hernandez-Losa, who is currently the director of the Molecular Biology Laboratory at Hebron Hospital in Spain. And the second presentation will be given by Dr. Ines Ranieri, who is a staff pathologist at the Institute of Pathology Medica AG in Switzerland. The first presentation will be about the NGS evolution in solid tumor genomic profiling. Javier actually will tell us a little bit his experience about using the S5-XL. And the second presentation will be actually on the first experiences using the Oncomine Comprehensive Assay Plus. With this, I also would like to remind you that right after these two presentations, we will have a Q&A session. And the speakers will be in the position of answering to your questions. For now, I would like to start with the first presentation. Please go ahead, Dr. Javier Hernandez-Losa. Thank you very much.
Hi, good morning. My name is Javier Hernandez. I'm currently the Molecular Biology Lab Director in the Hospital Vall d'Hebron in Barcelona, Spain. The title of the speech is The NGS Evolution in Solid Tumours Genomic Profiling. This is my disclaimer. And then I divided the speech in two different parts. The first is our diagnostic laboratory NGS experience. And then the second part is the most clinical research part of the NGS that we are running right now in the last 1 year. So first of all, who we are. We stay here in a big hospital in Catalonia, in Barcelona. It's Vall d'Hebron. And in Vall d'Hebron, you can see the Department of Pathology. There are 3 buildings with more than 20 specialist pathologists, including in the lab, and also a research institute just behind to perform different experimental designs. So in the experimental branch, we have a molecular lab. This is a translational molecular pathology lab. The chairman and the department is Dr. Ramón y Cajal. And also, beyond of this clinical research and translational research, we have a molecular pathology lab. These molecular pathology labs, there are at least currently more than 30 different molecular techniques, plus 25 in situ hybridization tests, and more than 10 RT-PCRs designed for clinical diagnosis. And we are also running over 16,000 samples per year. In addition, we are not only do the analysis for the patient from our institution, but also in Spain, we are on a reference lab who are receiving samples for other hospitals beyond of that. And then also for different ways, we have reference centers with different companies. And also the main idea is to standardize the molecular techniques in those samples, perform some efficacy in the samples management, reducing the turnaround time in different biomarker testing, and finally, to be cost effective in the consumption of reagents. So in different biomarkers, we have established different algorithms. Mainly, we receive samples for non-small cell lung cancer, colorectal cancer, and melanoma samples. And in base of this algorithm, we can develop different analysis based on immunohistochemistry, based on FIS, or based on PCR or qPCR. Finally, in some of them, we are using NGS to perform more broadly analysis in the same samples. As a reference lab, we always stay deciding which is the best technology in each biomarkers. On one side, we have a clinical validated analysis and a standard criteria to be positive or negative analysis by FIS or immunohistochemistry. But also we incorporating additional tests such as NGS, RT-PCR, LDT-assays, or nanostring, for example, with a more objective interpretation. We have covering most potential usability of different rearrangements. But in contrast, we have a clinical validation pending, and also there are no standard criteria to use those analyses. So as a result, why we use NGS analysis? From my point of view, the NGS analysis are more related in three different aspects. One is accuracy diagnosis, such as soft tissue tumors or pediatric tumors, biomarker testing, or translational research. First of all, in the soft tissue sarcomas, in our center, we have a clinical center who are collecting not only criteria for pathologists, but also for the clinicians. We have selected as a reference center in soft tissues in Spain. So based on the morphology data, inclusive within the specialist pathologists, there are also 25 or 30% of the cases that are discordant cases, because it's based on the experience of these pathologists and the expertise of each of that. So as a result, the molecular analysis, based majority of them based on rearrangement, maybe is worthy to incorporate this molecular analysis in order to solve these discrepancies. Why we use NGS and how to deal with the rearrangements? There are two main ways. One is DNA-based or RNA-based. From our side, we prefer RNA-based in order to avoid some large introns, repetitive introns, or very complex genomes events between the exons for the different genes involved in this rearrangement. So in our experience, we are using a platform for NGS in order to adequate and to characterize the right rearrangement. So we are covering unknown rearrangement. And also, this is the presentation with a software specific for describing these rearrangements. In the bottom part, you can see the cosmic presentation for the transcript that we discover. And also, additionally, in some cases, we are validating with orthogonal techniques, such as FIS, at least, for example, in some, if we have available for these props. In our experience, we are running 91 samples. There are 52 no fusion, but 36 is fusion, and only 4 invalid results due to the low quality of RNA extracted. In our experience, the majority of the Ewing sarcoma and alveolar sarcoma and other tumor types were different rearrangement described in the P chart. The most important thing here is that at least 8% of the samples were currently identified by NGS, and some of them has modified the original version of the diagnosis versus the final version of the diagnosis. In our case, we have an internal molecular pathology board. So this is one per month, 48 hours before sequencing results. This is an internal meeting with the specialist pathologists who are ordering the different tests. And also in this meeting, we have the specialist pathologists and also molecular biologists to discuss all the molecular analysis beyond of the NGS, FIS, or qPCR, or whatever, in order to define what is the concordant or discordant results with the morphology. Safe to the non-small cell lung cancer, in our experience, this is the workflow that we have established for different samples to arrive at our lab to perform the molecular profiling in this tumor type. We are cutting 8 slides and staining the first and the last one, such as hematoxylin in order to confirm the tumor content in each of these slides. And also the 2 former is including an extracted DNA for EGFR mutation by single gene approach. ALK, GROS, and PD-L1 testings are screening by immunohistochemistry, and in some cases, if we need to confirm our FIS analysis, we're using the last extra slides. So why NGS in non-small cell lung cancer? One of the reply is to perform a molecular profiling in these tumor types. In this review, we can identify, so the early stage or the metastatic stage, the different alterations that we can find in those tumor types. And as you can see, EGFR, KRAS, BRAF is the most important one, but there are many, many other alterations, such as amplification and rearrangement, to be very, very important in this molecular profiling. To do that, the best way is to choose the gene panels. So at that time, you need to define what is your interest. If you are interested in the class 1 actionable mutations only, or you can go to the class 2 for the clinical relevant alterations, or move to the class 4 in order to describe more discovery in these tumor types. Once you have established your intention, the second option is what is the technology that you are using, or amplicon site or hybrid capture. So both of them have pros and cons. You need to analyze in your site what is your pros and cons. In the amplicon, you are defining very well your amplicons, but on the other hand, you have more error in amplification and sequencing. Having said that, at that time, in our site, we decided to move to a panel who incorporate class 1 actionable mutation, and also combining DNA and RNA from the same samples in the same assay. So in this case, we decided to use Oncomine Solid Tumours, who are developing these genes to include it for the DNA mutation, and also we are performing rearrangement for ALK, RET, CROSS, and NTRK1. And also, the most important thing also is that there are very few amount of DNA or RNA as an input in this assay. Importantly also is it's not only for the known fusions in those genes, but also through the imbalance that this is provided for the kit, you can also recognize another alterations involved in these genes with a partner that are unknown. So we use the Ion Reporter. This is the software provided by Thermo to use this analysis, and applying by different filters, you have discovered the variants that you will find with those samples. In our case, we are starting with a different type of material. We have started with different samples for surgery samples, for cytology samples, for small biopsies, and also introducing some external artificial control. And also we are using a different type of DNA extraction and also RNA extraction. So the idea was to know exactly what's going on with these panels in our plethora of samples arrival to the lab, and also comparing these, the gold standard analysis, so such as qPCR by EGFR mutation, immunohistochemistry, and FISH with the OST. In these panels, we observe on a 100% concordance in this alteration that we have observed previously in terms of EGFR mutations. And also, as you can see in the table, in the last part, we found also additional alterations that are not covered at the beginning because we are comparing the data only for the gold standard, such as TP53 mutations and KRAS mutations and other alterations, very, very important in this tumor type. So in our experience, we have analyzed more than 176 samples, and this is the alterations that we found in our experience for the research. So this is the tail for the cancer, where discovered the most frequent alteration is TP53, following by KRAS and EGFR, as described in other series of other hospitals. Well, at that time, we are not alone in the community. So based on the results that we are observing in labs within the Europe and out of the Europe, there are some organization in terms of the coordination in the lab, performing NGS analysis mainly. And more importantly, in the France experience from the Institut National of Cancer, the INCA models, they designed specifically 28 laboratories to perform NGS biomarkers testing with a high level of quality criteria and terms for the equity in the whole populations. We tried to do this in our country in Spain several years ago with unsuccessful results. But moving forward in this molecular world, we decided to start a huge project, at least in this community in Catalonia, with 7 hospitals that are coordinated by ICS, the Institut Catalán de la Salud. The main idea is to shift from the current technologies that I explained before, immunohistochemistry, sequencing, FACE, or qPCR, for the next coming technologies in some of these 7 hospitals. The next coming technology are using NGS, are using digital PCR, and also nanostream to perform not only molecular studies in terms of research, but in a future, why not, incorporating our armamentum for the pathology report. This is the ideal world. So one reference lab within this project as a sex for different technologies in order to choose what is the best technology in terms of whether the biomarkers that you are planning to map. And also not only for paraffin samples, that is our gold standard, but also why not? The next one is liquid biopsy. Our recent clinical research and NGS experience is, okay, we acquired in our lab also the Ion S5 plus Ion Chef, and the reason was mainly to perform this testing biomarker in non-small cell lung cancer and present some data before, and also in agreement with another laboratories within our institutions. As an example, for example, we are in contact with our pediatric research group in order to start in some projects regarding how implement this NGS analysis in our lab and not beyond for the standard criteria right now. We are running some projects. We are using whole exome sequencing in some high risks for pediatric tumor, but we need to externalize those samples for a company. And this is 30 days later, they back the results. And then we need to move for a quicker way in order to understand if it's worthy or not, and we are planning to incorporate the childhood here. And also, for example, in agreement with hematology, they are running some research as a myeloid panels, and they are using this equipment. So the idea is not only acquire the equipment for our installation, but also to sharing this as a core lab here. And also, within this project that I mentioned before for the X project, we took advantage to incorporate Genexus. In this acquisition, the main reason was to optimize the workflow. So we have at least the workflow established in the 4 samples per lane. So this is a very flexible workflow for us. There are a full automation. So for us, this is very essential because this is not dependent for the high value of the technicians here in my institution. We are reduction on the turnaround time of the orders, and also possibility to combine the paraffin samples and the future liquid biopsy that we want to incorporate in our labs. So to do that, this was the main project, but of course the COVID infection and the pandemics arrived. And also in Spain, it arrived as a tsunami. It's not a wave, it was a tsunami. So at that time, we have different challenges. One is the installation. There are some accessibility to the lab. There are some reduction. We need to install the Genexus, they install the software, and also set up the machine. Having said that, there are a special attention for the engineer team here that Jaime Vicioso is a special thanks here because he stayed with us to set up during the whole process. But also there are some WebEx or some software that are solving part of our initial problems, such as LifeLens or Rescue software. So The first approach was to use samples that we had used previously by NGS analysis and run with the OPA, the Oncoman Precision Assay. In this table, you can see that the main alterations was the same like we have expected to find such KRAS, BRAF, P53, EGFR, etc. But also the most important thing is that when we compare exactly the same analysis with the previous method by NGS, not only found the same alteration that is expected, but also the allelic frequency variance was very, very similar. So here you can see that the deviation is very, very low. So as a result, and also to summarize, we are using OPA for this previous analysis, and in our research also we found a malignant mesenchymal tumor as an example that we performed different immunohistochemistry and we found the NTRK expression. We use the OPA analysis to know if it's NTRK fusion or not, and we observed that the NTRK fusion in this part, we observed an imbalance in the NTRK3. So thanks for the OPA analysis, we can confirm that the NTRK was rearranged in the NTRK3 gene. Here, to final my presentation, I would like to present you a short video, less than 3 minutes, in order to know and to show you what is the workflow for the next coming month when we incorporate the OPA in our analysis for the starting for the arrival samples to the end. Well, so having seen this video, my take-home message for this presentation is that, at least in Spain, NGS are broadly used in oncology with great experience in clinical trial, prescreening, monitorization, and translational research. That NGS is implementing in clinical practice with small and moderate panels focusing hotspot mutations and rearrangement. And also, finally, the technology improvement and the increasing automation process, such as Genexus, are essential for the future incorporation of NGS technologies in pathology lab and its use in different tumor types beyond the non-small cell lung cancer, but probably next coming tumor types are essential to be mapped by NGS. So final question, this is the lab for the molecular lab for the people who are doing this NGS. And also thanks for all of you for your attention.
So hello everybody. Well, first of all, I would like to thank the organizers of the meeting for inviting our institution to give this talk at this virtual meeting. And I'm Ines Ranieri. I'm a pathologist, also trained in molecular pathology. And I'm happy to present to you our first experiences with Oncomine Comprehensive Assay Plus for comprehensive genomic profiling. I do not have any conflict of interest to disclose, except that a honorarium is provided for the presentation. Now, to give you an introduction to the topic, I just would like to briefly go over a timeline of some of the highlights of precision oncology research. Probably the precedent is the treatment of estrogen receptor positive breast cancer with tamoxifen. However, the first truly molecular targets were also in breast cancer, HER2 amplified breast cancer, which responded to trastuzumab, and also the treatment of BCR-able translocated chronic myelogenous leukemia with imatinib. Later, it was found that EGFR mutated adenocarcinomas of the lung responded to TK inhibitors, that KRAS mutated adenocarcinomas of the colon responded less well to EGFR inhibitors, and that melanomas with BRAF mutations, D600, responded very well to BRAF inhibitors. To this point, basically, one was looking at single genes in very distinct entities. However, thanks to genomic profiling of a lot of different tumors, mostly by TCGA, it was found that a lot of tumors actually shared common drivers, and the idea of tumor-agnostic therapy evolved. In tumor-agnostic therapy, you don't anymore look so much at where the tumor is from and what the histology is, but you look at the molecular and phenotypic profile of the tumor. Today, there are two truly tumor-agnostic approaches, which are FDA approved. One is the treatment of all MSI-high tumors with immune checkpoint inhibitors, irrespective of whether they are from colon, from endometrium, or from any other site. Also, in 2019, it was approved that NTRK-fused tumors, irrespective of whether they're childhood, adult carcinomas, or sarcomas, could be treated with specific inhibitors. If we are looking at the oncology technologies and the requirement landmarks, it's certainly NGS massive parallel sequencing which led the foundation to our current genomic profiling of tumors. And without NGS, we wouldn't be where we are today. And indeed, several platforms have been developed and are in the meantime approved by the FDA and also for Medicare and Medicaid patients. If we look at the advantages of sequencing panels, now we save material, which is frequently very limited. Always consider that we are frequently dealing with very small biopsy specimens and cytology samples. Next, we save time. Before sequencing was often done in sequential order. When we did Sanger sequencing, for example, in adenocarcinomas of the lung, it was first tested whether mutations in EGFR or KRAS were present, the two most frequently mutated genes. And only if these were negative, then we would perform Sanger sequencing of other genes. Third, there is a growing number of actionable targets, and we just couldn't actually efficiently address these anymore by Sanger sequencing alone. And then lastly, because it was, as already discussed, we know that there are recurrent genomic aberrations across very different tumor types. The workload has increased, and we can look in very rare tumors whether there might be some target which could be relevant for the patient. Now, if we look at OncoKB, we find that actually we have at the moment 42 genes for which an FDA-approved drug is present. Now, these 42 genes, of course, could be easily covered by a small sequencing panel, but tumor mutational burden has started to be of interest because it is one of the parameters which basically predicts whether a patient might respond to checkpoint inhibitors or not. High tumor mutational burden in breast cancer and colorectal cancer is actually approved for checkpoint inhibitor therapy. Also, microsatellite stability is an issue. As already mentioned, this is a true tumor agnostic therapy. Of course, we easily can perform immunohistochemistry for MMR proteins, but sometimes we don't even have enough slides to do that, and we will have to rely on a DNA-based test. And lastly, there might be only 42 FDA-approved genes, but if we look at my cancer genome, we find actually that there is an abundance of clinical trials, and at the moment, there are over 9,000 phase 1 to phase 4 trials which look at very different targets. We think that this graph actually outlines very nicely why it's good to have a wide sequencing panel. This is by Vega et al. Sanchez from 2018 from Cell, and they looked at the 10 most frequently mutated pathways in cancer. Now, if you just take the receptor tyrosine kinase RAS pathway, you see that the two most frequently mutated genes are KRAS and BRAF. However, you might have mutations in rare genes such as SOS1 or ARAF, and they also might be potentially interesting and might be potentially targetable. And if you have a small panel, you just lose these. Actually, these authors have found that within these 10 pathways, you have 51% of the patients they screened had one target present. And if they expanded that to BRCA and to IDH1 and 2, it was 57%. So you have actually a considerable number of patients where you find a target if you use a broad sequencing panel. Now, genomic profiling in clinical research, and on a very small scale, that's probably what we do or how our laboratory evolved has gone on in many other places. We started out doing HER2 immunohistochemistry in fish in breast cancer and gastrointestinal cancer patients. We moved on to Sanger sequencing in, for example, lung cancer or colorectal cancer. And as our workload increased, we started to do small targeted gene panels, Oncomine focus assay. And soon it became obvious that clinicians were interested in researching wider panels, especially BRCA, but also tumor mutation load, and we started to apply these panels. And we decided now to try to implement the Oncomine Comprehensive Plus assay in our laboratory. In this assay, we have over 500 unique genes which you can profile for mutations, for fusions, and for copy numbers. And in this talk, I only will talk about the DNA part of the test. But you also have a multi-biomarker profiling looking at microsatellite instability, tumor mutational burden, but also mutation signatures. You have a really low DNA and RNA input as far as 20 nanograms, which helps a lot if you're dealing with small samples. You have a high sequencing rate, and you have a dedicated bioinformatics suite, which leads to a reduced TAT for reporting and which can be applied in a routine laboratory. For our setup to look at the assay performance, we actually started out with 21 FFPE tumor samples. In 16 and in between there are actually 17 samples. We know microsatellite status either by immunohistochemistry or PCR. In 8 samples, we had previously performed tumor mutation load assay, and in all the samples, of course, we had detected variants, SNVs, and copy number variations. If you look at our samples, you see actually that we have quite a variety. We had 5 adenocarcinomas of the pancreas, we had colon, lung, but we also had rare cancers, such as one carcinoma of the adrenal gland or a poorly differentiated neuroendocrine carcinoma, which was not further specified. And we actually chose the samples based on that we either had tumor mutation load data or we had a high number of, or a relatively high number of variants to compare. If we look at MSI status, that's how we routinely do it. We do either MMR immunohistochemistry. You see here an adenocarcinoma of the colon staining for all MMR proteins that are usually screened for, and or we do MSI PCR using the Bethesda panel, and in addition NR21, NR24, and 27 markers. So we're actually looking at 8 markers. Now, comparing our data to OK Plus data, we had 2 cases which were MSI high, and both these cases displayed a loss of MLH1 and PMS2 by immunohistochemistry. In one case, we also have performed PCR, and this case was MSI high with loss of all 8 markers, or shifts of all 8 markers. In the microsatellite stable cases, and I also would like to mention the cutoff is 20. Our highest null loss was 9.4, so it was well below the cutoff. We had no loss of MMR proteins by immunohistochemistry in all except 2. In one, we didn't have results because we didn't have slides to do it. We only had DNA. And in one case, we had an indeterminate result by immune, and we thought it is a false negative result from PMS2, and we did PCR, and it was actually turned out to be microsatellite stable. So we had, in all the cases, we had a match between what we had found with immune or PCR and the results from OK Plus. Also, looking at the MSI scores of all the cases, you see here the 2 MSI high cases well above 20, the 20 cutoff line, and all MSS stable cases were well beyond 19, which would be the cutoff for a MSI low score. And we didn't have any indeterminates so far. Assessment of tumor mutational burden. We had 21 samples. In 13, we only had results from OK Plus. In 8 samples, we had previously determined tumor mutational burden with the Oncomine TML assay. And you see here the samples which we had results from tumor mutational load assay and from OK Plus. And you see actually what we think is a really nice agreement. We have 2 cases that are high. Two samples that are high. Sample 11 is an adenocarcinoma of the colon, and sample 3 is a desmoplastic melanoma. We had two samples that were intermediate, and this one is the only where we have a slight difference. 10 is considered a cutoff for tumor mutational burden high, and this was 9.7 and 12.3 in the two assays respectively. However, we think that there won't be a biological difference, and we are also well aware of the fact that absolutely can compare TML assays in between samples we performed. The other samples basically distributed as follows. We had 4 samples which were in the intermediate range, 1 sample, an adenocarcinoma of the colon, microsatellite unstable, that was high, and the other samples were low. And we wanted to correlate our TMB high samples with the MSI status. And sample 3 is a desmoplastic melanoma, is microsatellite stable and has a high TMB. And sample 8 and 11 are MSI high adenocarcinomas of the colon. If we look now at sequence data, we looked at MLH1, PMS2, MSH6, and MSH2. And you see that case sample 11 here had an MLH1, had an MLH1 premature termination in codon 319. Sample 2 didn't show any mutations in these MMR genes. However, it showed a PV600 mutation in BRAF, and this is of course well known in the literature that these tumors exhibit hypermethylation of the MLH1 promoter. This desmoplastic melanoma sample did not show a BRAF V600 variant. However, it had a BRAF variant. I will just come to that. And also, it didn't show any variants in these mismatch repair genes. This is now the desmoplastic melanoma, the histology. You see very high tumor content. And if we look at the variants, you see that we have a NF1 frameshift mutation. We also have frameshift mutations in FANK-A and CDKN2A. In addition, we see this BRAF variant in the glycine of 469. This presumably corresponds to a class 2 BRAF mutation. And we have a HRAS variant in glycine 13. And these are actually uncommon variants for desmoplastic melanomas, which have more frequent, or for melanomas, which have more frequently BRAF variants V600 and NRAS variants Q61. If you look at tumor mutational burden, the signature, you see that you have a UV signature here with a really high amount of CT transitions. And our case actually, or our sample actually fits very nicely, which was described in the literature by Eruglu et al. And they did whole exome sequencing of 17 desmoplastic melanoma samples. They found in all of them a very high tumor mutational load. And they found over 82% of CT transitions, UV damage signature, what we have. And they found that these tumors have NF1 mutation as their genetic driver in the absence of the usual BRAF V600 and NRAS Q61 hotspot mutations. So basically, our case fits very nicely into this group. Variant calling in the 28 assays we performed before. This is Oncomine focused, comprehensive, and TML. We actually had found 57 variants in 29 genes. And with OCA Plus, we had 4 out of 57, 5 were false negative. twice a CDKN2A, once a CDKN2B, and 1/3 promoter alteration. Now, this would give a sensitivity of only 92%, and I have to mention here that we had some childhood problems, we might call it, at the beginning, and we are seeing now better results. Also, we did a Serosec control with the 7% DNA, and we had a perfect match with that. If we look at CNVs, that's certainly our biggest problem at the moment we are facing, in that we could call CNVs in 17 of 21 samples. However, in less than half of those, or in more than half, we had actually an acceptable MAPD value, which was below 0.4, but in slightly less than 50%, it was higher. And we are thinking about actually moving on to a different DNA extraction protocol. Sometimes we also get tissues from other institutes which have been fixed in formalin for a very long time, and of course we cannot control for that. If you look, we had copy number gains in 10 out of 17 samples, and in 15 out of 19 we had correct calls. If we look at the false negatives, we had 3. You see that were all low-level amplifications of once CCNV3 and FLT3. So high-level amplifications, we had no problem, and we were in all cases concordant. Also, there was a false positive Richter, which is also a low-level copy number alteration. In summary, we had highly reproducible determination of MSI by Oncoman Comprehensive Assay Plus if we compare the results to our immunohistochemistry and/or PCR data. The TMB results, which we determined by Oncoman Comprehensive Assay Plus match the results previously obtained with the TML assay, and also the values correlated with the biological and/or genetic characteristics of the tumors. For example, the MSI high tumors were both displayed high tumor mutational burden, and also the desmoplastic melanoma as described before. Variant calling, we had 53 out of 57 were concordant, and we had, as already mentioned, the 3 false negative if we compare the data to OCOV3 and 1 compared to the TML. I have to mention here that this is this TET variant, and it had very low coverage already in the TML assay. Now, what are the advantages and disadvantages? If we look first at the disadvantages, we think that the pre-analytical steps are even more crucial if you do a large sequencing panel compared to small sequencing panels. And of course, the analysis is more time consuming. However, the advantages is you have a really fast turnaround. You don't have to send out the tissue with the consecutive loss of time. The TMB can be evaluated. You do not have to combine assays anymore. You can evaluate the MSI status without requesting that you have a non-tumor control DNA. We don't always have that for samples. And you can analyze a wide array of genes which play a role in pathways important for oncogenesis and which actually may be relevant. Now, what are our plans? We really would like to improve our CMV calling, and as already mentioned, there's this improvement of pre-analytical steps, and the goal is call amplifications more reliably and also call deletions. We haven't actually started that yet. And we would like to compare our results in 2 samples of which we have F1 results. And I would like to thank all the people who work with me in this team. It's Constanza, Francesca, Noemi, Shirin, Martina, and Lenka. They work in the laboratory. They're really great. And then Simone, she is also a molecular pathologist, and the head of the department is Davide. And I really would like to thank all of them, and thank you for listening.
All right. So thank you very much again, both the speakers. Thank you, everybody, for joining us in this event today. Now we will move on with the Q&A session. It's a couple of questions for the speakers. I'll start actually with Javier first. Javier, first, thank you again for your presentation. You mentioned that in your institution and your laboratory is working as a kind of a reference center, therefore also obtaining samples from outside your own hospital. The first question is, when you're working together with pharmas, especially in the context of clinical trials, can you tell us a little bit more about how stringent and, let's say, how important is for them turnaround time?
I think that, Luca, is very, very crucial, the question. Thank you for the question. So only for clarification, I'm working with pharma, but I'm working for the pharma is coordinated, the accessibility for the samples to our lab. It's not for entering in clinical trials, or we are performing standard of care biomarkers such as KRAS and RAS-BRAF or EGFR and others in lung cancer. So, of course, the turnaround time is very crucial here, because you take into account that once the reference oncology or pathology for the external lab who are ordering these tests, they are at time that we are not controlling in the lab. So we are at time for selection the best specimen to send out and also to need to add the time for the courier, transportation, and also arrival here maybe 4 or 5 days later. And we started our first day for the turnaround time. As you can imagine, that we need to feedback as soon as possible if they are in front of the patients and are claiming for the results for the treatment. Yeah.
Thank you very much for your question. The second question is as well for you, Javier, and you showed some data where you actually comparing fusion detection rate between different technologies, for instance, FISH or NGS. We also take the opportunity to have Ines as a pathologist with here so I will also expand the question to her. How do you see the future of FISH? Do you see that NGS it's kind of taking up more and more space, so to say, because you can detect multiple fusion at the same time? Or do you still see FISH as, let's say, the hardcore standard, and NGS it's like on the top as confirmation? So what do you do in Vall d'Hebron?
I think that this is the opposite. No, I think that the NGS could be more discovery and also provide you the information in order to follow these alterations. But probably the FISH could be more standard, the criteria for the level of alterations. For example, ALK or ROS1 is established for 15% of the tumor cells are positive in order to be positive. And also in NGS, you can see present or absence, or maybe you need to count the reads, also performing for this specific transcript. So I think that the both of them are very, very useful. But probably in the future, next generation sequencing with these technologies who are developing new fusion panels, discovering new fusion alterations with partners that are not previously known. But at the end of the day, the split analysis by FISH usually is confirmatory.
Yeah. All right. Thank you very much for that.
I don't know if Ines agree with me.
Exactly. I want you guys, Ines, what's from your point of view as a pathologist, what do you think, how things will evolve, let's say, in the near future?
Well, I think, I mean, if we have NGS, for example, RAS fusion or ALK, we do FISH confirmatory. We don't do FISH anymore upfront. Okay. I mean, the only place where we do actually FISH at the moment a lot is breast cancer, because there you don't go up front, of course, with NGS, but I think that basically, for screening, that basically is not done anymore with FISH, like for example, lung cancer, because the panels are so good and they are so reliable, and so you really don't need it. And often FISH sometimes on tissue can be really hard.
Absolutely. And actually, this is a question for you also on something that you just have mentioned. From the audience, they are referring to your copy number variation. And you also just mentioned breast cancer, and you usually look for HER2 amplification and whatsoever. what do you see, again, the future in here? Do you think that it will be also the case that a lot of those testing will be replaced by the ability of NGS to detect copy number variant?
Well, basically, usually you have a HER2 test, at the beginning of the diagnosis, and at the moment, the clinicians, of course, they don't want to have a huge molecular analysis. They basically want to have like 4 or 5 predictive markers, and that's it. And I think for this, we will just stay with FISH. But I think, for example, we just had like a metastasized carcinoma of the salivary gland with ERP2, and basically these can be easily screened, and there we won't do upfront FISH, and I think there it will go away. That's what I think. But I think for breast cancer, as long as the therapy is what it is, and that's basically trastuzumab or estrogen receptor therapy or anti-estrogen receptor therapy, you don't need a panel. You need a panel once the patient has metastasized.
Right, absolutely. Thanks for that. Now another question for you, Ines. That's a little bit of a provocative question from the audience. And, we have heard the presentation, which was a little bit more on targeted sequencing with mid-size, more or less, panel. And then we heard yours, which was a little bit more about large gene panel, 500 genes, which has TMB, MSI, and all together. How do we combine those two words together? So in other words, if you look, for example, at the ESMO guidelines, which are referred here in the question, how would you say, and then also, Javier, please feel free to chip in also in the question. Would you say you go for a standard mid-size panel for everything, and then for more, let's say, complicated cases, or for example, cancer of a non-primary, then you will go for a very large gene panel? What is your approach? Maybe Ines first, and then Javier afterwards.
Basically, of course, the clinicians decide. We don't decide for them what they are doing. So, and what we are seeing is that usually they go for a large sequencing panel if they have an unknown primary, if they have young patients with metastasis, where they really are desperate to find some target, and where you basically also don't have guidelines, like for colorectal cancer or lung cancer, you have really good guidelines, but there you don't have guidelines. And that's basically where they are really doing these large panels. Also pancreatic cancer, they're often very interested in large panels because these patients basically don't have a chance, and you just really look, do they fit into a trial? Can you move them into a trial where they might benefit?
Absolutely, makes a lot of sense. I think, Javier, I'm not sure if you're still there. Your image froze.
I'm staying.
You look very suspicious in the videos, but please go ahead.
I was shocked. No, it's a joke. Yes, I'm still here. Yes, of course.
All right. So, what's from your side? Do you agree with what Ines said? We need to find, let's say, the right balance between large panel and a more applicable, so to say, mid-size panel?
Yeah, I think that I fully agree with Ines in this system. So I think that our criteria was to focus in the small panels in order to be more accurate in the small alterations, and they are also crucial following the guidelines. But also maybe there are some patients that you need to map or to understand more broadly the molecular alteration behind of these tumors. And also we extend in another institution here in Bio with panels from 300 genes or something like that in order to know exactly more in detail, more potential background of these tumor types. But initially we are focusing in small moderate panels.
All right. Thank you very much again. We have one very, very last question. We have only 1 minute to go. And it's actually about the Genexus. You showed this actually the beautiful video as well. Thank you very much for that. The question is about, in your first, let's say, experience in this first few months that you got it, is how flexible the instrument it really is. Have you managed to try to use it all in a single lane or multiple lane? How was your experience with that?
Yeah, absolutely flexible. So for our first approach, because we have a small experience with Genexus we acquired recently, and also we combine different lines, and also within the different lines, also different programs, such as only DNA, only RNA, DNA plus RNA, and also combining different samples. So the flexibility for our side is pretty good. So we have also good quality in the library preparation and the reads. And also from our side, in our short experience, it's quite promising.
All right, thank you very much. That's actually very good to hear it from, again, real life experience. So now I would like to close the Q&A section. Thank you again, both of you, for participating, for your presentation, but also for taking the time to answer to this question. I also would like to thank the audience for joining us today and remind them not to leave us. The event is still going live. You can check all the previous presentation. Please also check our posters hall. And also, in about a minute from now, we will have my colleague Andrew, who's actually live from our laboratory in California. He will do a demo about the Genexus integrated sequencer. You can take the opportunity to ask him a question. I'll be very happy to answer that. And also take the opportunity to get in touch with my colleagues, expert, NGS expert that are waiting for you in our chat room. Once again, I would like to thank you all very much, and I'm looking forward for the next event very soon. Have a great day.
Bye.
Okay. Thank you, Luca.
Bye.